We are the paradoxical ape. Bipedal, naked, large-brained, long the master of fire, tools, and language, but still trying to understand ourselves. Aware that death is inevitable, yet filled with optimism. We grow up slowly. We hand down knowledge. We empathize and deceive. We shape the future from our shared understanding of the past. CARTA brings together experts from diverse disciplines to exchange insights on who we are and how we got here. An exploration made possible by the generosity of humans like you.
Thank you, everyone, and I'm glad to be back at CARTA. I was here a few years ago, and I'm going to give you a little bit of an update on our research. So I thought I would kick us off by talking about some major questions in human origins. So these include things like, was there a single ancestral population in Africa from which the Homo sapiens lineage emerged? Or were there multiple human populations that all contributed to the formation of our present-day species? When did this happen? Was this 100,000 years ago, 300,000 years ago, 800,000 years ago? Where did these ancestral populations live? And were there any genetic adaptations that facilitated our subsequent expansion across the African continent and out of Africa? So I'm going to be quite ambitious today and try to answer 3 of these questions. Questions 4 and 5 are extremely difficult to answer. I have been working on them for a while. So I'm a population geneticist, which means I study human genetic diversity across global populations. And one of the things that we do is we reconstruct the demography or the history of the species. And we do this in a variety of complex mathematical models, and we parameterize them. So I wanted to give you an example from about a decade ago of a standard human population history model that my colleague and I put together using a publicly available data set. So you can see that this is probably incomprehensible to most of you. That's okay. I'm going to walk you through it. It's incomprehensible to most of us as well. But what I want to point out is that the populations that we are sampling, from which we are inferring these demographic parameters, are the Yoruba, who are up top, so they're from Nigeria, Europeans from Utah, just here, and Chinese and Japanese populations. So these are these standard populations that were initially sequenced in the 1,000 Genomes Project just over a decade ago, and from which we've obtained a lot of information about human history. When we first looked at these genomes, and my co-author Simon Gravel and I started thinking about what sort of models to put together, the focus was very much on non-African populations. So you can see that the Yoruba here are kind of a block. We have an ancestral population, and then they grow, so the width of this block increases, and then they just basically remain static for the next 150,000 years. So there's nothing else happening to this population. And that's because the focus was really on modeling this out-of-Africa bottleneck, which is this long, thin line here, the divergence between European and Asian populations, how much gene flow is occurring, and et cetera. I was very unhappy with this focus, but that's what was going on at the time. And so I had started working in Southern Africa. I have a field site there that I'll talk to you about. And I had this burning question, which was, can we really represent human demographic history when we're only looking at a single African population? Is a single Western African population from Nigeria sufficient to really inform us about what's going on in terms of our human origins. So this is a different representation of human genetic diversity. This is what's called a principal components analysis. And what you need to know is each dot represents a single human genome, okay? And the closer two dots are together, the more similar these individuals are. The different colors here represent different populations. So, for example, in these boxes, colored in blue, are Western and Eastern African populations. So you can already see that they have quite a bit of divergence between them. The Western, who are in the navy blue, and the Eastern Africans in green, they've already pulled apart from each other. There's already genetic divergence between them. And then let me highlight the out-of-African populations, East Asians, South Asians, and Europeans. They all kind of cluster together in this orange box. They have reduced genetic diversity because we all went out of Africa, or they went out of Africa and went through a bottleneck, so they lost lots of alleles. So they tend to be more similar to each other, in fact, than even Eastern versus Western Africans. But then you might notice there are all these other colored dots going on the graph. And the question is, who are these people? So these people are individuals that we collected in South Africa, and many of them carry ancestry which is related to their Khoe-San progenitors, so the indigenous people that were living in Southern Africa. And the first few boxes I showed you, those are all part of public databases, but these South African samples were not. So there's this entire axis of variation that was completely unsampled and unknown until we really began working there, we and others. So I think, to answer this question, can African genetic diversity be represented by a single modern population? Absolutely not. You need representation from multiple regions in Africa. And I'm going to talk to you a little bit about why I think the Khoe-San are so special. So this is just to give you a little bit of a geographic representation of where I work. I've been working in Southern Africa since 2005 amongst many different populations. We started in the Kalahari Desert with the Khoe-San population and expanded out to other groups. Everybody in our sample carries some Khoe-San ancestry from these hunter-gatherer and pastoralists that were living in Southern Africa when Europeans arrived in the 1600s. So if you've heard the word Khoe-San before, you might be thinking these are Bushmen or a San. You might have an image of your head of a man with a bow and arrow or something like that. But the people that we work with today are fully modern individuals that have been absorbed into modern day lifestyle. So many of them are farming individuals, they're shepherds, they work for wage labor, they dress just like this, just like you or I would. So I want to make very clear that these are fully modern individuals that we're working with, and they have their own concerns. I'm going to use the term Khoe-San to refer to members that are both San, which is an old term for hunter-gatherers, and Khoe, who are sheep and goat and cattle pastoralists. We now know from the genetics that these two groups of people have a similar genetic origin, so I prefer to sort of lump them together and call it Khoe-San ancestry. So sampling can look something like this. And so this is from our first sampling expedition in 2006 when we were working with the Khoe-San. And you can see that actually the target individual that we're sampling is hidden behind the tree here to protect her privacy. I'm taking notes. My translator is just next to me. And then her family is basically sort of looking on. And I love sampling in this context, even when it's really slow, because then the family has an opportunity to ask questions, to make objections. So there's a whole process of consent that goes on when you take DNA from human individuals. We have to be very careful about this, but luckily so far I've had a really wonderful experience working in Southern Africa. It is a slow process, so sometimes we do more group-oriented sampling. It stresses me out, though, to try to consent 30 people in 6 hours or something, so this is not the preferred method of doing it. We work all across South Africa and into Namibia, and sometimes you'll just be driving down the road, you see somebody walking. This is a shepherd in the Cedarburg Mountains. He has his whip right here, which is line underneath the consent form, and my project manager just pops out of the car and sits down next to him and says, can we talk to you about our research? Would you be interested? And he's really gracious and says, fine. And this is my last images of my graduate student, Dana, sampling a man in the Karoo while our research assistant, looks carefully over her shoulder at the paperwork and any signal that the man might not be comfortable. So if you're wondering what it's like to collect these DNA samples, it's an incredible experience, but it has to be done very, very ethically and very carefully. Okay. So let me get back to sort of the theory side of this. I presented to you initially this really simple model of the West Africans diverging from Europeans and East Asians. There was this out of Africa bottleneck. Okay, this is fairly standard stuff. But what is going on within the African continent over the last few 100,000 or million years? So there were a couple of very common models, and my colleagues at Davis, Teresa Steele and Tim Weaver and I put together this review paper on them. So I'm gonna walk you through them. And the first one is just the simplest model. It's a single origin, so all humans within Africa and outside of Africa have a common origin, which is relatively recent. So this would be our common origin, and then humans diverge into southern, western, eastern Africa. Europeans and everybody else come off of this East African branch. Then there was another model, sort of more recent, in 2018 and so forth, in which people introduced this idea that there was archaic admixture in Africa. So everybody's probably familiar with the idea of archaic admixture from Neanderthals and Denisovans outside of Africa. But some geneticists then posited this was also happening within Africa, and in fact disproportionately affecting Western African populations, even though we don't really have good fossil remains of who, hominin-wise, was living in Western Africa during this time period. So these were the two sort of main models. And we tried to flesh it out by thinking about other models derived from archaeology and paleoanthropology. And these include, yes, there were older populations living in Africa, but they sort of got subsumed by this recent expansion. So it's kind of a blend of the single origin model with these older divergences, but they were all Homo sapiens. And then this last one, which was put out by colleagues, called African multi-regionalism, which is the idea that this substructure, this divergence between different African populations is quite deep within Africa, goes back 300, 400, 500,000 years. So it's a very, very deep divergence. It's not shallow like this first model. Now, in order to try to adjudicate between these different models, you need evidence. And being a geneticist, I decided to focus on what I do best. So I went back and teamed up with my colleague from 2011, Simon Gravel, and one of his postdocs. And they had developed a new computational tool to handle very large amounts of genomic data. And we summarize this data using something called summary statistics of different types of components like linkage disequilibrium. And then we were able to collect additional samples from Eastern, Western, and Southern Africa. So those include populations from Ethiopia, like the Amhara, Oromo, and Gamuz, the Mende from Sierra Leone, and then I was able to contribute a Khoe-San population, the Nama, who live in South Africa and Namibia. So now we have representation from 3 different major regions in Africa. And then we added in the British, just for fun, and the Neanderthal as well, to kind of look at the divergence from an archaic specimen. This is the largest sequencing data set of Khoe-San genomes, at least to date. So it was a really, really special opportunity to combine all of the samples together. And I'm skipping over like all of the technical details. So you can look at this slide and then think like 2 years of technical details, and then we'll move on to the results. And this is the demographic model that we inferred. So it's sort of similar in style to that first gray model I showed you. It's just sort of been flipped on its head. So this is 1 million years ago, and this is present day. we're representing these different genomes that we sampled by populations down at the bottom. So here are Southern Africans, Western Africans, Eastern Africans, and Europeans. Okay, and then our Neanderthal gene is just up here. So each of these letters here represents a different parameter that was inferred in the model. And these parameters are things like migration rate or divergence time. So migration rate is really a way of saying, like, how frequently did genes move from one population to another population across some structured geographic space. It's impossible, well, hopefully not totally impossible, but quite challenging also to read this model, so I'm going to try to break it down for you into slices. So we're going to start at the oldest part of the model. So this is roughly 1 million years ago, probably more like 900 to 1.2 million years ago. And what we find is that indeed there is very deep structure contained within modern human genomes that goes all the way back to 1 million years. So we've labeled this structure population stem 1 and stem 2, representing these stem populations that contribute to everybody's genome, okay? And what's really interesting is that we find there is very consistent migration between stem 1 and stem 2 for a long period of time. So for hundreds of thousands of years, there are two populations in Africa. We don't know where they're living, but they're exchanging migrants back and forth and back and forth. Okay, we call this a structured population in human genetics. Neanderthals appear to be more closely related to stem 1 than they are to stem 2, suggesting possibly stem 1 is closer to Eastern Africa, very, very hypothetically. Then we say what we call a reticulation in the model, or this breaking apart and coming back together of these different stems. So stem 1 seems to split into two more stems. So then we have stem 1e and stem 1s. And this occurs roughly around 400,000 years ago. So still quite deep when you're thinking about human prehistory. And then they basically remain isolated from each other for several 100,000 years. So we have 3 different stems, these, if you volume the cursor, these 3 ones, that are isolated from each other for several 100,000 years. And this is super interesting and also puzzling what sort of traits and cultures developed uniquely in these 3 stems during that time period. Does that help explain differences in archaeological material that we see in different parts of Africa? Really, really interesting questions. Then we also see another major event, which begins about 120,000 years ago. And this is right coincident with some major climate change, which is occurring in Africa, called MIS5. So about 120,000 years ago, these stems begin to merge together. So they go from this period of isolation to this period of merging. And what happens here is then sort of the formation of the modern contemporary African populations that we see today. So stem 1s and stem 2, you can see this orange and this red color, merge together to become the ancestors of modern-day Khoe-San people like the Nama. This stem 2 and the stem 1 merge, and they contribute to the modern-day ancestors of West African populations. Stem 2 and stem 1 merge. These are the East African populations and everybody else. Remember, people who live outside of Africa have so little genetic diversity, they're almost a nuisance parameter, so they're just kind of in there. But that's everybody who has parentage from outside of Africa comes from this joining of stem 1e and stem 2. And that's consistent with a divergence between Eastern African and Western African populations around 60,000 years ago, followed by an out-of-Africa bottleneck. So what are the implications of this model for archaeology? I get asked this question a lot. It's really difficult to answer. I will say that there is archaeological evidence for increasing cultural innovation or an acceleration of cultural innovation during this time period, 100 to 120,000 years ago. So maybe this is coincident with these populations expanding and merging back together. It lines up well with the genetics, but there are a lot of additional questions that one could ask. I see I'm just running out of time, so I'm going to end by making three summary statements. So what we infer using these new genomes from Africa is what we call weakly structured stem, or two different stem populations that have been exchanging migrants for a very long period of time. This is in contrast to Homo sapiens versus Neanderthals, which are incredibly isolated during this period. So this lack of differentiation between stem 1 and stem 2 then allows us to make a prediction for the fossil record, that these hominins that were on either stem are actually not very morphologically dissimilar from one another, and we don't see any evidence for an archaic hominin admixing into these African populations. And then the Khoe-San, who are southern African indigenous populations, remain the first population to sort of isolate from these other groups, and we can date that back to about 120,000 years ago. I'll briefly thank my three major collaborators on this, my lab at UC Davis, our collaborators at Stellenbosch University in South Africa, and to all of the communities in Africa that contributed their DNA to the project. Thank you.